Okay, so in this lecture, we're going to look at this vignette on Gibbs sampling to fit a mixture model. So it's this one here. The idea here is that it's motivated again by kind of clustering genetic data. And if you remember our examples where we were trying to classify elephants as being forest or savanna elephants based on their DNA. Okay, so it's very similar to that, except that now we don't have any reference populations, reference samples that tell us what the allele frequencies look like in the forest group and the savanna group. So we've just got a bunch of elephants and we're told there are two groups here, essentially the forest and the savanna. And your job is to kind of classify the elephants into the two groups, or cluster them, maybe we should say, into the two groups. Okay, so it's kind of similar to that problem, but where we don't know those reference frequencies. So the idea is that we have to kind of jointly estimate the reference frequencies and the assignments, the classifications from the data. And we're going to do that by Gibbs sampling. It's basically a mixture model. So we're going to fit a mixture model by Gibbs sampling. So here's a setup. You've got genetic data on each of n elephants at R locations, and we're going to assume again for simplicity that elephants are haploid. That is, they just have one copy of their genome, which is not true. They have two, but just to simplify it, that means we can just label them as 0 or 1 and treat everything as Bernoulli, which makes it a little bit easier to code up and to focus on the main points. Okay, so there are two alleles at each locus, two types. We're going to label them as 0 or 1. So each individual's data is just going to be a binary vector of length r. Okay, there's an r loci, and so that I'm going to let xi here denote the genetic data for each individual. So xi is a binary vector of length r. I'm going to let big X be the collection of all the genetic data. So those are our data. And then just as we usually did, or as we often did, for example, in the fitting EM algorithm to mix, using the EM algorithm to fit mixture models, we're going to introduce these Z's as our latent variables. So Zi is going to be the unobserved label for individual I to say whether they came from forest or savanna. And so that's what the Z's are. And then we've got the allele frequencies. So for each of the two groups, we're going to have a frequency of the one allele in each at each locus, at each position. Okay, so for each group K, there's going to be R allele frequencies, one for each position in the genome. Okay. All right, so that's a notation. X is the data observed. Z is the classifications or the cluster labels or the component labels in the mixture model, unobserved. And P are the allele frequencies for the forest and savanna groups, for the two groups, also unobserved. Okay. Okay, so to do Bayesian inference for Z and P, which is what we're going to do now, we need to specify the likelihood, P of X given Z and P, and a prior distribution on Z and P. Okay, and then we're going to be able to, then we're going to use Markov chain Monte Carlo and specifically Gibbs sampling to sample Z and P from their posterior distribution given X. Okay, so the likelihood is given here. It's basically exactly the same as in that very first example. Given Z and P, the idea is that the X's are draws, Bernoulli draws from the relevant allele frequency. So given that you know which population it came from, and given that you know the allele frequencies, you're just drawing Bernoulli, independent Bernoulli data at each position. Okay. So that's the likelihood. And in our prior, we're going to assume that P and Z are independent. Okay, so P, remember, is the allele frequencies, Z are the allocations. So if I tell you the allele frequencies, before you know the data X, just telling you the allele frequencies doesn't tell you which sample came from which group. So it's reasonable to assume Z and P are independent. And furthermore, we're going to assume that for the P's, the allele frequencies are independent at each position along the genome, which is a simplification. It's not necessarily true. If things are very close together on the genome, there might be correlations, but say simplifying assumption that they're independent. So we've got some prior on the P's that are independent. We're also going to assume that the origin of each individual is independent. And we could allow there to be unequal balancing of the two groups. So we could say, well, we don't know what the frequency of the two groups is, and introduce a pi parameter, for example, and estimate that jointly with Z and P. But for simplicity, I'm going to assume that we know for now that the two groups are equally represented in the sample. Okay, again, that's just for simplicity to get the ideas, make the ideas easier to grasp. So essentially, Z is going to be, each Z is just going to be a toss of a fair coin, 0.5001. Okay, so just with that in mind, we can maybe I can write down the graphical model. So let me switch to my iPad. Yeah, so just let's draw a picture of that. So we were having the Z1, Z2 up to Z. There was a Z for each Z. So we're going to draw the graphical model for what's just gone on. We've got the P's down here. I'm just going to write a big P here. And then X1 are the data for individual one. So if we knew Z and P, we could, or sorry, if we knew Z1 and we knew P, we would be able to generate an X1. And if we knew Z2 and P, we would generate an X2. So some people find it useful to draw out the graphical model here. It's not entirely necessary because all that we really need at this point is to compute the conditional distributions we need. But sometimes it's helpful to have a graphical representation. So in particular, we can see from this that the joint distribution P of X, Z, and P is, well, the Z's don't have any parents, so it's Zn, and the P doesn't have a parent, so we can P, and then we have P of X1 given P and Z1, dot, 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 P of Xn given Zn and P, right? P and Zn. So let me correct that. That's the same, but it's a bit confusing to switch. Okay. All right. So that's hold that thought. That's the joint distribution. Now to implement a Gibbs sampler, I'll stick here. We want to sample, right? P of X and sorry, P of Z and P given X. So the Gibbs sampler iterates the following step. So sample P of Z given P and X, and sample from P given Z and X. Okay, so those are the two steps that if you iterate those from our intro to the Gibbs sampler, you should see that you're going to, from those two conditionals, you're going to get a Gibbs sampler whose stationary distribution is this joint distribution. Okay, so all we now need to do is work out what those conditionals are. And the vignette takes you through the first one, and the second one has a gap that you'll be invited to complete. But let's just look at this here while we're here. It's useful to notice that well, firstly, it's useful to notice that this factorizes, right? Because okay, and also that it's always true that the conditionals are proportional to the joint. So this is always the first thing that I write down when I'm deriving these conditionals is to say they're proportional to the joint by Bayes theorem. You know, basically, you know that because they're equal to the joint divided by what you're conditioning on, right? By Bayes theorem. right? So they're proportional to the, so it's always true for any condition, it's always proportional to that joint, but it's important that the proportionality here is, means as a function of z, right? As a function of whatever you've got before the condition. So the constant of proportionality, the normalizing factor, doesn't depend on z. In fact, as you can see, the normalizing factor is that. But we're not going to worry about what the normalizing factor is. We just know that it doesn't depend on z, right? Okay, which and as we've already written down that joint. So the nice thing is that every conditional now use the same joint to do it. And basically, you look at that joint and you ignore the parts that don't depend on z, right? Because and in particular, in this case, you can see that they factorize. So that's a kind of a key observation, is that the terms that involve zi don't involve the other z's, right? So the first thing, so because this conditional distribution factorizes, it means you can just work out what each of the independent conditionals are and then multiply them together, right? If the conditional factorizes, then yeah, they're independent a posteriori. So so we can now just focus on a single zi, which actually is given xi, right? Which is proportional to, well, as we can see, p of zi, p of xi given p and zi. And actually, now you should see that we're back to that original problem that we tackled in, I think, class 1, which is to work out what's the probability that a task or a sample came from a given population, given the allele frequencies p and its genetic data, right? So we're back to that 2-class problem. What's the posterior probability or the conditional probability that it's a savanna elephant versus a forest elephant given the p and the x's? Yeah. So another way of thinking about this is that first step is exactly the 2-class problem, the Bayesian 2-class problem that we started the class with. Okay, and then the second step turns out, we won't do the details here, but it turns out that that's just the estimation of an allele frequency based on samples from a population, which was also an early vignette that I don't think we covered in class, but there's a vignette on estimating. Basically, if you remember, if you put a beta prior on p and you get samples from the group, you know which, because we're conditioning on z, you know which samples came from which group, so you're going to get a beta posterior for p here too, conditional on z and x, as long as you use a beta prior. So so these two problems are kind of simple. Maybe I can just say again, just as before, p of p given z and x, which we're trying to compute now, is still proportional to that joint, right? But now it's not, it doesn't mean that it's the same distribution as for z, because now the proportionality is as a function of p. So the constant is not going to depend on p, but it will depend on z, right? So now when you're working out what this is, you want to go back to this conditional and ignore the terms that don't depend on p, right? So in fact, you can see these terms depend on p, whereas the others don't. So you can see that it's proportional to p of p times the product over I of p of xi given p and zi. And you can now start to factorize that in terms of the loci, right? So actually, if this factorizes over loci, I can't remember whether I used j for loci or some other symbol, but let's use j now there. So that's let p of j be the data at locus, the allele frequencies at locus j. Of course, there are 2 of them, but let's ignore that for a moment. And then this also can be a product over J, right? And in fact, when we do that, we'll see that the graphical model we wrote out was actually too granular, if you like, because in fact, if you think about P being, if we think about splitting P into P1 up to P L, these are the these are the L loci, then actually the data, x1 1 up to x1L only depend on the relevant P. The data at position 1 only depends on the frequencies for position 1. Of course, we also need x2 because it's for the next individual L. Also depends on those. So drawing out these graphs, there are actually various notations that people use to kind of get around the problems I'm having here with with repetition basically and fitting everything on a page. So if you're interested, you can look up, I think there's some ideas called plate notation, where you draw boxes around these things to represent replicates. Anyway, okay, so back to this, just to sketch out what this is going to look like. For each k, we can actually factorize these over k by introducing the alternative representation of the z's as a binary indicator for whether, instead of having zi is 1 or 0 for each k, have a zik be 1 if I came from population k, that alternative representation. You can write this all out in terms of products, and this this will have a beta prior, or each pjk will have a beta prior, and these terms will be binomial terms, and you'll get a beta posterior, because beta is conjugate for binomial, for each pjk. Okay, so I'm not going to do the details, but that's you need to derive that. And so if I stop my share there and share my go back to the vignette here. Okay, so the idea is that that's exactly what's implemented here. As you can see, there's a gap here in the derivation that you can try to fill in. But let's just have a look at what the Gibbs sampler code does then. So this is a function for computing the likelihood, basically. And this is a sampling code to sample from z given p and x. And this function samples p given x and z. And if you see, here we go, there's a beta distribution here that you can derive. And so the Gibbs sampler here just iterates between sampling p and sampling z, sample p, sample z. So think of it, it's kind of an iterative process where you estimate the allele frequencies, and then assuming that you've got them right in some sense, you estimate, quotes, estimate. Think of the sampling as kind of a fuzzy or a noisy estimation of the labels. And then given the noisy estimation of the labels, you re-estimate the allele frequencies, again, using a kind of a fuzzy estimation. When I say fuzzy, it's because you're sampling, so you'll get a different answer every time just because of this sampling. And what the Gibbs sampling theory tells you is that that fuzziness is exactly right, right amount to get the posterior distribution to get. So there'll be uncertainty in the estimates of p, partly because there's uncertainty in estimates of z, and every iteration your estimate of z will be slightly different. And so that uncertainty gets propagated, is one way of thinking about it, through to your uncertainty in p. Okay, and so if you, this is a visualization of the formation of the Gibbs sampler. So each row here is represents a sample. So the first sample is the bottom row, and each sample, you know, and basically each column is a elephant, and the color represents which. This is an image of the Z's, right? Image of the Z's, and so it shows which group each elephant was assigned to in each iteration. So we started with a random assignment of individuals. I should say I simulated data here where there were two groups, and you know, under the right model. So the Z's were randomly assigned, and there were two groups with quite different allele frequencies. So, and then I initialized the Gibbs sampler with a random guess for everyone's assignment. So that's a pretty bad guess initially. So you can see here, this is, you know, this is the table of like, how well did we, you know, this is the Z on iteration 1. So this is how, you know, misassignment, as you can tell from random guessing, you wouldn't expect to get many correctly assigned, or half of them on average. So that's what you see in the initial assignment. But by the 100th iteration, it's settled down, you know, to getting most of the, well, okay, have they got most of them right, or it's got the most of them wrong? It's assigned most of the ones that were really from group 1, it's assigned to group 2, and most of the ones it was assigned to group who were really from group 2 were assigned to group 1, and that's something called a label switching problem. So the idea is there's nothing to tell, nothing in the data to tell you which one should be called group 1 and which one should be called group 2. So this is actually, you know, if you think about it, this is clustering them pretty much correctly. These 4, you can think of these 4 individuals as being misclassified from the clustering point of view, because they should have been put with these, with well, with one of these, probably with this one, I guess. Okay. So again, you can, and this is kind of like seeing how the iterations progress over time. And what you can see is you only need a very short amount of burn-in before the MCMC starts sampling from something that looks like it's, you know, looks like a sensible part of the posterior distribution from a random starting point. Okay, so that this is kind of, think of this as a kind of a trace as you go up here, a trace of what the MCMC thing, MCMC Gibbs sampler is sampling from.